In the realm of biology, the fascinating concept of biological immortality is often discussed. This refers to a state where an organism's mortality rate doesn't increase with age, as seen in certain unicellular and multicellular species, including some vertebrates. However, it's important to note that biological immortality doesn't mean invulnerability to other potential causes of death, such as injury, illness, predation, environmental changes, or resource scarcity. Biological immortality has spurred debate in the scientific community. For instance, the Handbook of the Biology of Aging queries the authenticity of this phenomenon, suggesting that although the mortality rate might stabilize in old age, it remains unusually high. The concept extends into cellular biology, where biologists use the term immortal to describe cells unbound by the Hayflick limit, a maximum to the number of times a cell can divide due to DNA damage or shortened telomeres. Alexis Carrel, prior to Leonard Hayflick's theory, even hypothesized that all normal somatic cells were immortal. The term immortalization was initially associated with cancer cells that express telomerase, an enzyme that lengthens telomeres and helps cells evade apoptosis or program cell death. Immortal cell cultures like the HeLa and Jurkat are extensively used in various biological research fields, from creating vaccines to studying cell metabolism. Besides, embryonic stem cells and germ cells are often referred to as immortal. It's also possible to render cancer cells biologically immortal through the induction of oncogenes or the loss of tumor suppressor genes, often done virally using the large T antigen. The concept isn't limited to humans and lab cultures. Many organisms in nature exhibit signs of negligible aging. For instance, the Greenland shark can live between 250 and 500 years. Recently, researchers at Alphabet's Calico have found potential evidence that naked mole rats don't suffer from increased mortality with age. Unicellular organisms usually age and eventually die. Yet, under perfect growth conditions, organisms that divide symmetrically can achieve biological immortality. Each cell division rejuvenates the cell. This concept is the basis for considering stem cells and gametes as immortal. The tiny species of jellyfish known as Turritopsis dohrnii, or Turritopsis nutricula, holds a unique power in the animal kingdom thanks to its potential for biological immortality. These 0-20-inch creatures owe their undying nature to the process of transdifferentiation, essentially refreshing cells post-sexual reproduction. The process can roll out eternally, theoretically granting the jellyfish infinite life. Bred in the Caribbean waters, time has seen to dohrnii disperse across the globe. Crucial to its regenerative capability are DNA replication and repair, and stem cell renewal, according to genomic research. However, these marine creatures are not alone in displaying such age-defying attributes. Similar phenomena have been observed in Hydrozoan Laodicea undulata and Styphozoan Aurelius spoon. Even lobsters, despite not having the traditional trappings of immortality, do not appear to age or weaken or experience fertility decline. Older lobsters may even be more fertile. Scientists attribute this to telomerase, an enzyme that repairs DNA sequences at chromosomal ends called telomeres. Contrary to popular misconceptions, though, lobsters aren't immortal. They molt to grow, a process that requires energy that continues to increase with the size of the shell until the lobster succumbs to exhaustion during a molt. In fact, older lobsters often cease molting altogether, resulting in deteriorating shells leading to their death. Another mention should be made for planarian flatworms, who reproduce both sexually and asexually. The Schmidtii mediterranea genus of these planarians is known to regenerate indefinitely. Their inexhaustible regenerative capacity is driven by a very active population of adult stem cells. Though both kinds of planarians experience age-related telomere length decline, the asexual individuals have the capacity to maintain these lengths, thanks to their regenerative abilities. This is in stark contrast to sexual planarians who restore telomerians only during sexual reproduction or embryogenesis. Sexually reproducing planarians rarely live beyond 3 years due to the fading ability of neoblasts to replace aging cells. However, asexual planarians have shown phenomenal longevity, with individual animals in some species living over 15 years.